Good morning. This is going to be the first of some series of genetics lectures. I hope you will find them helpful. I'm going to send all of this material to you as PowerPoints. The PowerPoints are almost certain to be more complete and probably more coherent than any kind of lecture I can do as a recording. So I encourage you to look at the PowerPoints whether or not you listen to the lectures. I think that will be important. There will be a set of 3 PowerPoints total. That may be a series of 4 or 5 lectures. I'm probably going to break the lectures up a little bit more so that they are smaller recordings and to make it a little bit easier for me to do them. We are going to be covering all of chapter 11. Chapter 11 is a cluttered chapter that you should consider looking through. I'm going to be covering a little bit more than you will find in your book. And then we will have some sort of genetics test, and also there will be the genetics problem sets to do, one that's definitely for homework and one to probably work out as you go through the lectures so that you can practice doing the types of things you need to for genetics and be comfortable with it. Now, the first thing about genetics is to put it in context historically. And obviously everyone has known since the dawn of time that offspring inherit their parents' characteristics, but you would probably be surprised at the enormous variation in what people thought was actually going on, and no one actually knew what was happening genetically, of course, for a long time. So that, for example, the Greeks believed, and this belief continued all the way through the 17th through the 18th centuries to some degree, and was considered a fairly scientific and objective type of theory at the time. But this belief was that each sperm cell contained a complete tiny human being that the male had created, and that the female served mainly as an incubator for the baby and did not really contribute genetically to the baby at all. To get around the obvious idea that the mother plainly put some characteristics into the baby, people tried coming up with different ways to explain that. And for example, that the womb itself, the uterus, imprinted characteristics on the baby, even though the mother was not actually passing on her own characteristics in any kind of real way. And the baby itself was created by the person, by the man. Obviously, this was not true. But it wasn't clear what was really happening at all. Mendel, as your book will explain, was the first person to really explain what was happening with genetics. Prior to that time, one theory was put forward by a person named Lamarck in France. His theory was that any type of organ or limb that was used more would contribute more to the development of an embryo, and that the progeny would therefore change to match their environment based on what the parents were exercising. This was wrong, but not stupid. Lamarck came up with ways that might work, all of which were mistaken, but all of which were coherent. A person named Lysenko in Russia really believed in Lamarckism, and this was during Stalin's era. And so when Stalin declared that Lysenko was correct, this caused Russian biology to have quite a problem. Lysenko believed that if you grew seeds in Siberia, the seeds would, because of the situation in Siberia, become better able to withstand cold weather. This was 100% not true at all. It contributed to famines in Russia, and it probably this type of belief set Russian biology back at least 100 years. It took a long time to recover from this, which is a good lesson to us today about the perils of politicizing science and why it's important not to do that, not to let that happen. Your book doesn't discuss that. Your book discusses Mendel and his pea plants. You should read through that section. I am personally bored with peas. We are going to talk about dogs more than peas because I think that is more interesting. But read through the pea example. The important thing about Mendel is that he was mathematically inclined and was therefore not just breeding one pea with another via hand pollination, but rather than saying a lot of green offspring appear and some white offspring, or a lot of yellow seeded offspring appear plus some that produce red seeds, Mendel was mathematically inclined and counted them and said, oh, look, we have an actual 3 times as many green to white, or 3 times as many yellow to red, or 1 yellow to 1 red, or 8 of these for 3 of those. And so Mendel was the one who looked at mathematical ratios and concluded that inherited traits must be inherited as little packets of discrete information, and that therefore inheritance was particulate rather than like pouring red paint into white paint and stirring and getting pink. The important thing for you guys to know is that inheritance is particulate, that traits are inherited as discrete particles. He called them factors, we call them genes, that inheritance is not like blending paint, generally speaking, that inheritance is particulate, more or less, generally speaking. And so we will talk about that. Now, we have 4 basic concepts which are important in this first bit, and they come in pairs. Here they are, right? So we have gene versus allele, and dominant versus recessive, homozygous versus heterozygous, and a genotype versus phenotype. And all 4 of those concepts are going to be important, and you want to really clarify in your mind what we mean by all of these. A gene is a discrete piece of genetic material on one chromosome, which carries a length of DNA, which is a length of DNA, which codes for a particular protein. That's what it is. A gene is a length of DNA on a chromosome that codes for a particular protein. Alleles are different forms of those genes. It's important to use those terms correctly. Alleles, the gene might code for hair color, and then an allele might code for dark hair versus blonde hair. A gene might code for cystic fibrosis, and then alleles would code for not having cystic fibrosis versus having cystic fibrosis. A gene might code for whether your earlobes are attached, and then one allele would be attached, another would be unattached. So the gene is the thing, and the alleles are the different forms of the thing. There's a lot of misunderstandings about dominant versus recessive. First of all, most genes aren't this simple in terms of dominance versus recessiveness. Second, dominance has nothing to do with whether a gene is common in a population or not, nothing at all. And also, dominant has nothing to do with whether a gene is good for you or not. Dominant genes are not good and recessive genes are not bad. All of that is a misunderstanding, and that's important, and we'll talk about that as we get to it, right? But those things are important to understand. Homozygous versus heterozygous, it's important to understand that homozygous means you have 2 alleles and they're the same. It doesn't matter whether they're dominant or recessive. If they're the same, you're homozygous. If they're different, then they're heterozygous, then you are heterozygous. Everyone has 2 alleles. Everyone has 2 alleles for every gene. Everyone has 2 alleles for every gene. If they're both the same, you're homozygous. If they're different from each other, you're heterozygous. The letters that you will be using to code for genes and alleles, that's your genotype. We'll talk about that as we go. Letters are your genotype. Your physical appearance is your phenotype. Your physical appearance could be black versus yellow in dogs. It could be dark-haired versus blonde-haired in people, right? It could be whether you're resistant to flu viruses and the Chinese virus, or whether you're not resistant to it. All of those things are part of your phenotype. You can think of it as stuff you see, but see can be a pretty broad term here. And we'll stop there just for a minute. Okay, we're back, right? Here we have dogs, right? We have the genotype shown with the letter and the phenotype shown with the words, right? You can see that if you are EE, you're black, and if you're E little e, you're still black. This is a black lab. In both cases, the lab is equally black, right? The letters are the genotype, blackness is the phenotype. If the dog is little e, it's yellow, right? The letters are the genotype. What it is yellow, which is the phenotype. Let's do this cross, right? You put the gametes along the top and the bottom, right? The big E, little e dog, that's the male, put it on the top. It can either put a big E into a sperm cell or it can put a little e into a sperm cell. Those are the choices you get. That's what goes on the top. The female is only going to put little e's into her egg cells. That's the only kind of E she has. Therefore, that's what she puts into all her egg cells. You get a Punnett square that looks like this. 50% of the puppies are black, 50% are yellow. You could say 50% are big E, little e, 50% are little e. You could say this is a 1-to-1 ratio. We would say that. It's a 1-to-1 genotypic ratio. It's a 1-to-1 phenotypic ratio. It should be obvious that labs come in black, yellow, and also brown. The extension allele, big E, does control black versus yellow. Other genes control the rest of it, right? For labs, the other gene that controls color is the brown versus black gene that works like this, right? We have the E gene shown on one side and the B gene shown on the other. Both of them code for black, or at least in the dominant form, they don't not code for black. And then the recessive condition with the E's is yellow and with the B is brown. This is an accurate representation of what's going on with labs, right? As it happens, in yellow, color is restricted from the hair so that the hairs are yellow. And in brown, color is restricted from the hair and the skin so that the hair is brown, the skin is brown, the nose is brown, and the eye rims are brown. You can tell whether a dog is carrying both yellow and brown because a yellow lab carrying brown will be yellow with a brown nose, whereas a yellow lab that's got black behind it will be a yellow coat with a black nose. Right? The phenotypes, right, are shown with blanks. This is a phenotype. It's a phenotype even though there's letters involved because there's blanks. We don't know what the actual genotype would be. What we're saying here is the dog is black because it's got a big E. And what we're saying here is the dog is still black because it's got a big B. You can definitely read it as black and still black versus opposed to this one, which is black, now it's chocolate, right? If you have a dog with this genotype, it's a chocolate lab. Pause the recording and do this Punnett square on paper. I'll give that a second. This Punnett square on paper. What gametes do the parents make? Where do you go from here? What kind of puppies can they have? What color are the parents? I hope you paused the recording and did that Punnett square. It would be good practice for you. You will see that you'll be doing a lot of things like this in your lab type problem set. And then of course, a lot more that are more complicated in the set to turn in for more points. Here's the cross that I asked you to do, right? Here's the gametes that the one dog can make. All he has is little e's, therefore all his sperm cells get little e's, right? Half of them get the big B, half get the little b. There's the other dog, all of her sperm cells, right, get the little b, and then half get the big, and then half get the big E and half get the little e. And then we fill in the Punnett square with the puppies. Okay? We get a 1:1:1:1 genotypic ratio and phenotypic ratio. Although half the puppies are yellow, some of the yellow puppies have a brown nose and some have a black nose. You can sort them out that way if you wish. Right? Therefore, we get 25% in each box that we can tell what they are. You'll be asked for a phenotypic ratio and a genotypic ratio in these types of crosses. Okay? That will do for this video. Go through the Intro to Genetics PowerPoint number 1, and it should go right with this material from this lecture. And you want to go through it carefully, pause it when you need to, do some thinking, work out Punnett squares when you're asked to, and make sure that all this material is clear before you go on to the second genetics lecture and PowerPoint.